Welcome to Bio with Aanchal. In this part of the presentation, we'll discuss about the different cell organelles. The part 1 of the chapter is already uploaded. In the previous presentation, we have already talked about cell, postulates of cell theory, cell size and its efficiency, then difference between prokaryotes and eukaryotes, and difference between the cell organelles and organs. Those who have not seen my previous video, you are requested to go into the description and you find the link there for the same. Now in this part, we will discuss about the different components of the cell. The living part of the cell, the non-living part of the cell. The main important portion for this is the cell wall and the cell membrane. Now the cell wall and the cell membrane, the difference is asked on the basis of their permeability. Permeability we will discuss little later. Then in the cytoplasm, what are the organelles present? ER, mitochondria, Golgi Apparatus, Lysosomes, Centrosomes, Plastids. We will discuss all this in detail. The non-living component like granules, vacuoles, fat droplets. In the nucleus, you have nuclear membrane, nuclei and chromatin fibers as living, and nucleoplasm, basically the fluid content, is the non-living. The next organelle is the cell wall. Cell wall is a unique structure present in the plant cells, and it is also present in bacteria and fungi. When you have to classify the living organisms, we can classify as those who contain cell wall and those who do not contain cell wall. So plant cells, bacterial cells and fungi, they all contain cell wall. In plant cell, the cell wall is made up of cellulose. In plant cell, the cell wall is made up of cellulose. Okay. In bacteria, the cell wall is made up of peptidoglycan. Okay. In bacteria, it is made up of peptidoglycan. In fungal cells, it is made up of chitin. So these are the three different components of the cell walls present in plant, bacteria and fungus. Now, different function of the cell wall, that is, it is a protective layer to all the plant cells. It protects, right? It provides mechanical support to the cells and determines the shape of the plant. The most important part is it is freely permeable. That means cell wall allows the entry and exit of all the substances into the cell. It is the cell membrane which decides that what should go inside the cell. Is that clear? Moving to the next, the cell membrane. Cell membrane is found between the cell wall and the cytoplasm in the plant cells, and it is the outermost membrane of the animal cells. It is generally composed of the lipoprotein, phospholipid and the protein. Lipo is what? Lipid. Proteins means you know that, right? Lipid plus protein is lipoprotein. Phospholipid is phosphate group and the lipid, that is fat and protein. It is also composed of carbohydrate and cholesterol. And the function of cell membrane is it gives assistance in maintaining the cell shape. It also holds cytoplasmic components, and it is a selectively permeable membrane. Now in your exam, difference between the cell wall and cell membrane is asked on the basis of permeability. So you have to write cell wall is freely permeable and cell membrane is selectively permeable or semi-permeable. Is that clear? Any doubt? Put it in the comment section. Cytoplasm, the jelly-like substance which occupies most of the cell, is the cytoplasm. It is present in plant cells and animal cells. It is the aqueous ground substance of the cytoplasm. It is also called cytosol. Okay, that is aqueous ground substance of the cytoplasm. The liquid part of the cytoplasm is called cytosol. And the living component of the cell is called protoplasm. Okay, the living component of the cell is called protoplasm. Protoplasm is basically cytoplasm plus nucleoplasm. Remember this, cytoplasm is what? In which all the cell organelles are suspended, and nucleoplasm is present in the nucleus. It is little denser as compared to the cytoplasm. And protoplasm is a living component of the cell. Only living cells contain protoplasm. Protoplasm does not exist in dead cells. Okay, so cytoplasm plus nucleoplasm is the protoplasm. It includes variety of cell organelles and other inclusions. What are inclusions? The vacuoles, the fat droplets. We have already discussed the non-living component. Then what is the function of cytoplasm? It is the medium for all the vital activities that is going on in the cell. It stores glucose, vitamins and amino acids, and it is helpful in exchange of materials between the cells and the organelles. So it also serves as, you can say, media of transport, the open sea from which the ship moves from one island to the other, transporting the material. Next one is the nucleus. Robert Brown in the year 1833 discovered nucleus in the plant cells. Okay, if you remember, Robert Hooke Robert Hooke discovered cells, right? Now, when Robert Hooke discovered cells, he coined the term cells also. He worked on the cork cells. Cork cells were dead cells. So, he discovered only the compartments. Okay, and he did not see the nucleus. So, he named them as cells. Okay, now nucleus is found in all the eukaryotic cells except the mature sieve tubes of the higher plants and mammalian RBC. RBC, sieve tubes. I hope you know what are sieve tubes. That is present in the phloem. And RBC of the camel, this is an exception. RBC of even some frogs and camel, they contain nucleus. But mammalian RBC, that is RRBC, has no nucleus. Why RBC has no nucleus? So that it has enough space. So it has enough space for oxygen. Is that clear? Now what does the nucleus contain? Nucleus contains nuclear membrane, nuclear sap, we call it nucleoplasm, nucleolus and chromatin fibers. Now nucleus is basically very large in size. You can see here, this is the nucleus, right? So inside the nucleus there is a denser material present which is called nucleolus. So nucleolus is generally smaller, present within the nucleus, helps in RNA synthesis and ribosome synthesis. So this nucleus is basically helping in RNA synthesis and it is helping in ribosomes synthesis. Okay. Nucleus is very large in size, bound by nuclear envelope, contains chromosomes rich in DNA, and nucleolus is generally, does not have a limiting membrane, just a denser material present inside the nucleus, and it does not have any chromosomes inside, rather it is rich in RNA. Is that clear? Any doubt, children? Put it in the comment section. Now next is plastids. Now plastids are unique organelles present in the plant cells. P for plastids, P for plant. So it is basically the unique organelle present in the plants. Okay. Now it is of three types: chloroplast, chromoplast and leucoplast. Now you can see the pictures of the chloroplast. Chloroplast is generally for photosynthesis. Chromoplast. Chloroplast is generally for photosynthesis. Chromoplast provides color to the fruits and the flowers, and leucoplast is for the storage. Now chloroplast, green in color, contains a pigment called chlorophyll. You have to remember this. Chlorophyll is a pigment which provides green color and chloroplast is an organelle. There is a difference, okay? Now what is the function of chloroplast? It contains pigment is not a function. It helps in the process of photosynthesis by trapping sunlight, that is the function, okay? And chromoplast, they are colored plastids other than green, and they are basically because of the presence of carotenoids, they are of different colors. Then leucoplast, generally it is colorless, it does not have any pigment, and based on the type of material it stores, the name is amyloplast, alleloplast and eleoplast. Now amyloplast is generally carbohydrate. Amylo, this word is for carbohydrate. Allero is for proteins, and the eleoplast is oil, that is your fat. Okay, that is, I am talking about the plants, children, here. Okay, in maize seed we have, I hope you remember, we have an aileron layer, right? Aileron layer. That aileron layer is generally the cells which contains aileroplast. Okay, which contains proteins which is required for the embryo to grow. Then these all plastids are interconvertible. They change. That is how now the chloroplast changes into chromoplast from a raw fruit to a ripened fruit. Next one, endoplasmic reticulum. Endoplasmic reticulum is a kind of framework around the nucleus. It is a network of membranes present in the cytoplasm. It was discovered by Porter and Thomson. Generally in ICC, the scientists are not asked, but for any kind of you can say Olympiads and all, you must be aware about these terms and these scientists. So Porter and Thomson in 45 discovered endoplasmic reticulum. They are present in all cells except the prokaryotes and the mammalian erythrocytes. Again here mammalian erythrocytes. When we were studying nucleus, we studied that the nucleus, the erythrocytes do not contain, that is RBC. It does not contain nucleus. Similarly, RBC does not contain endoplasmic reticulum also. So that RBC can squeeze through the capillaries. If endoplasmic reticulum is present, it provides a network, a framework to the cell, a shape to the cell. So the cell the RBC does not contain endoplasmic reticulum, so it provides it flexibility to move through the capillaries. Okay, they are made up of three components. Endoplasmic reticulum. Endoplasmic reticulum is made up of three components, which are the cisternae, the vesicles, and the tubules. Okay, you can see here there are two types, smooth and hard. As I said, cisternae and vesicles and tubules. You can see cisternae are the tube-like structures. Then vacuoles are the small round-like structures, and tubules are the larger tubes. Smooth endoplasmic reticulum helps in fat synthesis, and rough endoplasmic reticulum, which contains it contains ribosomes studded on it. It helps in protein synthesis. Now, to discuss the functions, ER serves as a channel for the transport of material between the various regions of cytoplasm and between the cytoplasm and the nucleus. Cytoplasmic framework to provide space for some of the biochemical activities. It is the endoskeleton of the cell, and it helps in the synthesis of fats, steroids, cholesterol, and protein. Now, fat, steroids, and cholesterol is for the smooth endoplasmic reticulum, and proteins basically the rough endoplasmic reticulum. Now, directly they are not involved in the synthesis. On the endoplasmic reticulum, there are ribosomes studded. These ribosomes helps in the protein synthesis. The proteins, it contains secretory proteins, and SCR plays a very crucial role in detoxification of drugs and poisonous byproducts. SCR is present in a very large amount in liver. Okay. Now what I was talking about here, let me just erase it. Or now what I was talking about, now you have an endoplasmic reticulum. On that you have ribosomes studded on it. Okay, the larger unit and the smaller unit. Now what happens, the what to say nucleus, from the nucleus the RNA comes and the protein synthesis happens. Protein is formed inside the ER, and properly it is then sent into a vesicle. This vesicle is transported into Golgi bodies. Okay, there all the processing and all happens. So proper folding and all is done in the ER, and final packaging and all is done in the Golgi bodies. Is that clear? Moving to the next slide. Golgi apparatus. The Golgi complex was discovered and first described under the name internal reticular apparatus by Golgi. The scientist named was Golgi, so the name was given to the organelle. Alternative names for Golgi complexes are Golgi bodies, Golgi apparatus, Golgi zones, Lipochondria, Dalton complex, idiosomes, Baker's body, and even dictyosomes in plants. Unit of Golgi complex are called dictyosomes in plant. This is a unique name given to the Golgi body in the plant. So you cannot say that plants do not have Golgi bodies. Plants do have Golgi bodies, but they are more complex. So the specific name is given to them, that is dictyosomes. Okay. Then they are also composed of lipids and fats mainly and are known as lipochondria. They are composed of cisternae, vesicles, and tubules, like what the endoplasmic reticulum also had. And it also has vacuoles. Now you can see here in the diagram the single membrane structure, this entire structure is called the cisternae, right? the cisternae, and finally they make many tubules. So this is all cisternae and tubes. They are all made up of tubes and cisternae, right? So this is your cisternae, tubes. These are small transfer vesicles, and the small structures which are round shaped, these are called vacuoles. Moving to the next, let us talk about the functions of Golgi apparatus. In exams, if the function of Golgi apparatus is asked, you need to write synthesis and secretion of enzymes and hormones. This will give you full marks. Going into detail of the function, secretion of enzymes and formation of packing of material for export, I'm sorry for this, for export from the cell across the plasma membrane by the process of reverse pinocytosis. Now, question for your children, what is phagocytosis and what is pinocytosis? Answer in the comment section. They form the plasma membrane of the cell. Anterior part of the sperm, that is acrosome, this is important. Acrosomes are formed by Golgi apparatus. This question can be asked in the MCQ. Then they help in the formation of cell wall in the plant cells. They help in the synthesis of various components of the cell wall and helps in the deposition and transfer of these components at the corner part of the cell. So it helps in the cell wall formation. Moving to the non-living substance present in the cell, the vacuoles. Vacuoles are the fluid filled or solid filled single membrane bound spaces. They are storage sac. This question is asked several times. What is the outer membrane of the vacuole called? Tonoplast. What is the fluid present in the vacuole? Cell sap. In plant cells, a single prominent very large vacuole is present which occupies around 50 to 90% of the cell volume. You know the plant cell, right? It has the nucleus shifts to the periphery. If you have a plant cell, you'll realize that the most of the component of the plant cell is being put to the corner and the vacuole takes the largest space of the plant cell. Okay. Then in plant cell, vacuole is bound by a membrane called tonoplast filled with cell sap. Cell sap, what is the composition of cell sap? It is rich in sugar, amino acids, protein, minerals, metabolic waste. It's not just water, it has many components present in it. Types of vacuoles, you have food vacuoles which are called phagosomes. You have contractile vacuole which is present in amoeba and paramecium, and the main function of vacuole is to maintain turgidity and rigidity in the plant and maintain the osmotic pressure in the plant. You have to write one or two functions, so keep this in mind. Next one is centrosomes. Centrosomes are the unique organelle present in the animal cell. Centrosomes are the unique organelle present in the animal cells. Plant cells do not have centrosomes. It's a unique organelle in the animal cell. It is found near the nucleus. It helps in the formation of spindle fiber during cell division. It helps in plant cells since centrosomes are not there. Spindle fiber originates in the cytoplasm directly. But in animal cells, you have centrosomes, and these two components of the centrosomes are called centrioles. You can see here, this one and this two, the tube perpendicular to each other, they are centrioles. It is not an organelle. They are together called an organelle. So if you write centrioles, you do not get marks for the organelle. If a question is write a unique organelle present in the animal cell, you have to write centrosome, not centriole. It contains two centrioles. Centrioles are the short bundles of microfilaments arranged at right angles to each other. Next one is the lysosomes. Lysosomes again are unique to the animals. They help in the process of digesting the foreign substances. It is also called suicidal bags. Now children, whenever a reasoning question is asked, you have to give a correct reason for any question. Now here the question is, why lysosomes are called suicidal bags? Because they kill themselves, they kill the cells in which they are present by releasing the hydrolytic enzymes when they find that there is some problem in the cell. So cell, if the cytoplasmic structure changes, if any kind of problem happens with the aging of the cell, The lysosomes release the content in the cell and digest the entire cell. That is why it is called suicidal bags. The main function of lysosome is intracellular digestion as it contains hydrolytic enzymes. Okay, it helps in removal of the worn out cells. This worn out cells matlab self killing. If the cell gets spoiled, the lysosome present within the cell kills the cell. That is why lysosomes are called suicidal bags. Mitochondria, very important. This will again go in detail when we are studying chapter 7, that is respiration in plants. The diagram of mitochondria can be asked to draw in the exams. It's very easy. Draw a sausage like structure, a bun, and just move with the boundary to make the inner cristae, that is inner membrane. This is how your mitochondria to look like. It is the powerhouse of the cell. It helps in the synthesis of ATP. Now, powerhouse of the cell is another name given to the mitochondria. But where function is concerned, you need to write synthesizes ATP. It is site of Krebs cycle and electron transport chain. Details we'll be studying in the next chapter, that is chapter 7, when we are doing chapter 7. Your question is, write the full form of ETC. What is the full form of ETC? In the comment section. The inner membrane of mitochondria is called cristae. It is a semi-autonomous body. Why semi-autonomous body? Because it contains its own DNA. The mitochondria and the chloroplast, they are both semi-autonomous because they contain their own DNA. Okay? And it is again absent in RBC. Why it is absent in RBC? To avoid the usage of oxygen. To avoid the usage of oxygen, that is to avoid the next respiratory processes. Okay, next one is plant cell animal versus animal cell. You know that plant cells have rigid shape, large vacuole. It is autotroph. Since plants are autotroph, they synthesize their own food. They have chloroplasts, lack centrosomes, lack lysosomes. Animal cells, the cell is irregular in shape because there is no cell wall. It has many small vacuoles. Vacuoles may or may not be present. It does not have chloroplast. It cannot synthesize its own food. It has centrioles and centrosomes and lysosomes. You can see here I have marked with the red the unique organelles. So in plant cells you have vacuole, large vacuole and cell wall as unique. Even plastids are unique. Okay, so you can have these all chloroplast. This is also a unique structure which is absent in animal cells. Okay, in animal cells you have lysosomes and centrosomes as a unique structure. Is that clear? Moving to the next, the prokaryotes and the eukaryotes. Now prokaryotes, it has got no true nucleus, and eukaryotes have got true nucleus. Right? It's unicellular. Eukaryotes can be unicellular and multicellular. Yeast and all are eukaryotic cells, but they are unicellular. And all the animal and the plant cells you can think about is a multicellular organism. All are multicellular organisms. Then eukaryotes can have both asexual and sexual reproduction, unlike prokaryotes which have generally asexual reproduction. Then the common features between them is both contain ribosomes, though they are different type of ribosomes. Both has DNA, cytoplasm, cell membrane. The structure is slight different. Next is this diagram. Your question is, find out the error in the diagram. Find out the error in the diagram. There is an error here. So your question is, find out the error in the diagram. Okay, you can have a look, pause, have a look, and then tell me what is the error in this diagram. Put it in the comment section. Moving to the next. Now you have to label it. Okay, ribosomes with the functions. So let me label it for you so that it will help you. 1, 2, 3, 4, 5, 6, 7, 8, and then only 9. Okay, and here you can say this one is A, B, C, D, E, F, G, H, I. Write the answer in the comment section. That is all for today. If you have any other doubt, put it in the comment section. I'll get back to you. Thank you so much, children, and happy learning.